So this chapter is all about information. And information in biology comes in layers. And it goes from cells up to at least organisms and in ecosystems and beyond. So information can include the information that's transmitted when a coyote urinates on a fence post and marks its territory, informing other coyotes that come by that this area is taken. And it goes all the way down to cells where, say, a hormone is transmitted from the center of the human brain at the pituitary gland to, say, the femur to stimulate bone cells to grow and cause a person to get taller. And it can go all the way down to the ultimate level, which is DNA, which is what causes proteins to get made, which in turn is what causes animals to urinate on fence posts or hormones to move around the body. So we're going to start out in this chapter with DNA, but before we begin, we have to make clear what the general principles of information in living things are all about. So the very first big idea here is ultimately this is a really important topic. This is one of our big ideas. Living things store, retrieve, transmit, and respond to information essential to life processes. So this is a really fundamental, foundational thing that all living things have and work with. And this information is heritable. In other words, organisms reproduce and they pass this stuff to their offspring. You know, whether it's hydras in a pond reproducing sexually or asexually, all reproduction involves, at the fundamental basis, the passage of DNA, or in some cases RNA, to offspring so that the offspring actually have the information to live. Generally speaking, the information that living things use is DNA. There are cases of a few prokaryotes, not many. Mycoplasmas, I believe, are one, and then viruses, which aren't even prokaryotes. They are non-living, disease-inducing chemical things. So most living things use DNA. Viruses and a few maybe cellular things use RNA as their primary storage material of heritable information. The idea is in that eukaryotes, heritable information is passed to the next generation through reproduction, and the cell cycle and meiosis, which we're going to talk about later in this unit, are important to passing down the chromosomes. Ultimately, that's what the cell cycle is all about, is trying to make sure the chromosomes end up in the right place so that they can do good work in a living thing. Ultimately, the chromosomal basis of inheritance allows us to understand how we pass information to our offspring. That's genetics. That was discovered by Gregor Mendel, and that was back in the late 1800s. Ultimately, though, don't want everybody to believe that Gregor Mendel's genetics explains everything. There are many inheritance patterns where basic genetics won't explain what's going on. And that's basically being explained by the new field of epigenetics, which is inheriting, instead of DNA from parents, it's inheriting control systems from parents that determines how your DNA behaves. But we'll get more of that later. So ultimately, genetic information is being always transmitted from parents to offspring. It's almost always DNA. It can be RNA. There's very little difference between DNA and RNA. We'll go through the differences later. Most living things use DNA. Some prokaryotes, very few, and lots of viruses use RNA. Some viruses use DNA. Do also be aware that there are some diseases where we don't really know what they're using to store and transmit biological information. That would be things like mad cow disease or chronic wasting disease in deer. Those diseases don't have a nucleic acid basis of information storage. So they're right now in the medical communities a bit confused about those things. A couple of other things: prokaryotes generally that use DNA don't have DNA structured like ours. Our DNA is basically like a shoestring laying on the floor, whereas prokaryotes, their DNA is more like a hula hoop the ends of their DNA molecules are connected to the beginning to make circles. So they have what are referred to as circular genomes. We have a linear genome. Our chromosomes are linear, and they have a beginning and an end. In fact, you may hear in this chapter me talk about telomeres. Telomeres are the ends of eukaryotic chromosomes, which are really important to the structure and the longevity of a chromosome. We also need to make you aware, just that for a note right now, but it's going to come up more later, plasmids are circular loops of DNA that are different than the circular genome of a prokaryote. Plasmids are tradable pieces of DNA, almost always, I don't know of any exceptions, almost always little circular loops of DNA that are exchanged between bacteria so that they can trade basic biological information. Something we can't do, something they can do. Now, what this graphic here is trying to tell you is that bacteria that's store their basic genomic information in a loop. Now, it's really not shaped like a hula hoop because it's all folded up in here, but it's a piece of DNA. It's connected end to start. In addition, they have little plasmids, which are much smaller, that they can actually shoot out of the cell into another bacterial cell. In fact, bacteria are quite fond of trading plasmids, and by doing that, it's sort of like people, like exchanging cell phones or exchanging cosmetics or something. You're giving each other tools to allow you to do new things. Generally speaking, on this planet, though, eukaryotic cells have linear chromosomes in here in the nucleus. This is the blue thing is a cell here. And inside of here, this is the nucleus blown up. We have these chromosomes, and generally speaking, chromosomes right before cell division look like X's because they are two pieces of DNA that are actually tied together in the middle by a little knot. And the DNA at the molecular level looks like this. It's actually, they call it a double helix. It's like a ladder that's twisted. So after this now, we'll move on to DNA research.